All right, so I'd like to welcome everybody this morning and thank you for attending our webinar on engineered reporter cell lines, advanced tools for profiling cell signaling pathways, and therapeutic target development. I'd like to introduce our presenter for the morning. That's Dr. Hyun Kyu Lee. He's the director of our bioassay development and screening team here at Novus Biologicals. He's been with Imgenix, which is now part of Novus Biologicals, for almost 5 years. Before joining Imgenix, Dr. Lee worked at Scripps Research Institute as a research scientist. All right, let's get started and welcome Dr. Lee.
Thank you, Cody, and I would also like to thank everyone for attending this webinar today. I'm going to go over our report cell lines, including mechanisms, usages, and applications. And later, I'm going to introduce cell-based drug discovery screening services we provide. A report cell line is an engineered cell line stably expressing the reporter gene under the control of target-specific promoter, which is designed to monitor cellular signal responses induced by stimuli. This is one example of the cell line stably expressing toll-like receptor 4 complex and also NF-kappa SEAPorter construct. When this cell line is activated by LPS, which is toll-like receptor 4 ligand, toll-like receptor 4 quickly propagates signal transduction, recruiting downstream signal molecules, and finally activating I-kappa B kinase. And I-kappa B kinase is phosphorylate I-kappa B and releasing NF-kappa B into nuclei and binding response elements, inducing SEAPorter protein. Express SEAPorter protein is secreted into cell culture media. We have two different types of reporter cell lines. One is SEAPorter cell lines, and the other one is LUCPorter cell lines. SEAPorter cell lines use the secreted alkaline phosphatase as the reporter protein, and LUCPorter cell lines use the granular luciferase as the reporter protein. SEAPorter cell-based assay, you can use any multi-well plate. You can plate SEAPorter cells in a multi-well plate for 16 hours, and then later you can treat the cells with agonist or ligands or agonist plus antagonist for 24 hours. And then you can measure SEAP from culture soup, and then you can analyze the data through SEAP quantification. LUCPorter cell-based assay is much more simple, and all you need is just one white plate. You can plate LUCPorter cells in white plate for 16 hours, and you can treat the cells with whatever ligands, agonist, or complex with agonist and antagonist for 6 hours or 16 hours. And then after that, you can directly add substrate into same white plate for 5 to 10 minutes, and then you can read the plate, analyze the data through luciferase quantification. We have 14 different SEAPorter cell lines covering toll-like receptor and the NF-kappa B SEAPorter targets. And then we also have agonist and antagonist products related to SEAPorter cell lines, including toll-like receptor ligands and antagonists such as NF-kappa B inhibitors, or MRD88 inhibitory peptide, or some TLR inhibitory peptide or compound. We have LUCPorter cell lines related to T-cell subset modulation study, including IL-17A, FOXP3, ROR gamma, gamma T, and GATA3, and NFET. We also have some agonist and antagonist related to those LUCPorter cell lines, including ROR gamma, gamma T plasmid for IL-17A promoter induction, and RONX1 for FOXP3 promoter induction. We also have antagonists and inhibitors, such as DPR66 is inverse agonist for ROR gamma, gamma T, and also celestrol, prolambazine, thymoquinone, also IL-17A inhibitor or ROR 410 inhibitors. We also have LUCPorter cell lines related to inflammation and cancer study, including AP1, STAT3, STAT1, and CXCL2, TNF-alpha, TNF-beta, and also we have interferon beta and ISRD. We have some agonists and also antagonists related to this LUCPorter cell line. Those LUCPorter cell lines and related product can be easily found on our Novus website. For example, if you click IL-17A LUCPorter cell lines, you can find application notes, and also together with those information, you can find agonist information, antagonist information. You can find also readout system information. We also have toll-like receptor stable cell lines. We cover whole mammalian toll-like receptors. And you can use these cell lines for TLR expression detection and calibration purpose or other multiple application. We performed cell line validation through many different ways. First of all, we did a cell number titration using two different formats, SEAPorter cell lines and LUCPorter cell lines. For SEAPorter cell lines, we used TLR8 SEAPorter cell lines using TLR8 ligand, which is rescue mode R848. And then we used 96L format assay, and the many different cell number titration and per wear. We optimized the cell number for this SEAPorter cell line. 50,000 per wear is optimal cell number for this cell line assay. We have LUCPorter cell lines, but we have different types of host cells for these cell lines. So we tried different types of host cell number titration. First of all, step 3 LUCPorter cell line uses HEC293, activate with IL-6. TR4MD2 CD14 LUCPorter cell host cell is HeLa, activated with LPS, and step 1 LUCPorter host cell is row cell 264.7, activated with interferon gamma. And same thing, we did many different cell number titration, and we calculate Z factor and S2B, and then we found out that the optimal cell number is 50,000 for HEC cell based, and 85,000 for wear HeLa cell based, and 100,000 per wear low cell based. We also did cell stimulation time analysis. For SEAPorter cell line, same thing, we use TLR8 using same ligand at 3 different time points, 6 hours, 16 hours, 24 hours. And then we found for SEAPorter cell line, optimal stimulation time will be 24 hours. In case of LUCPorter cell lines, we use TLR4MD2 CD14 cell line and activate with LPS. at 3 different time points. And then we see similar, pattern, 6 hours, 16 hours, 24 hours. So the optimal stimulation time should be at least 6 hours and go up to 16 hours. We check the DMS tolerance and SEAPorter cell lines, and same cell line we used and same ligand. And we tried different concentration between 0.1 and 10% DMSO. And then we found DMSO tolerance less than 2%. Same thing, LUCPorter cell lines, we use the same TR for LUCPorter cell lines and different concentration of DMSO, and showed similar tolerance rate and less than 2% DMSO. If I summarize, cell number titration for HEC293 host cell-based cell lines, 50,000, HeLa cell 85,000, and raw cell 100,000 per well in 96 well plate format. And stimulation time will be SEAPorter 24 hours, LUCPorter 6 hours, 16 hours. DMSO tolerance is less than 2%. And also we check the substrate incubation time for SEAPorter, 30 minutes to 60 minutes, and LUCPorter cell lines, you need only 5 to 10 minutes substrate incubation. If I explain cell line application, I divide into 3 categories: application, information, and effector. Effector is the molecule you are looking for. Targets are specific target for that molecule. You can use LUCPorter cell lines or cell lines you can for the study signal transduction pathways. The effectors can be kinases, phosphatases, modulators. The targets can be receptors, adapters, mediators, transcription factors. You can identify molecules such as ligands, agonists, inhibitors or antagonists, or inverse agonist. The targets can be receptors, kinases, phosphatases, transcription factors. Or you can validate molecules using LUCPorter cell lines. The effectors can be modulators, agonists, antagonists, or antibodies. The targets can be specific protein, specific molecular phosphorylation, or induction activation, or cell proliferation, or apoptosis, or specific protein expression can be targets. You can use LUCPorter cell lines for drug target screening, and there should be multiple effectors for the targets. And it can be an agonist, antagonist, inhibitors, ligands, or inverse agonist as effectors. And the target, we focus on major human disease, including cancer, inflammation, autoimmune disease, or cardiovascular disease. Related molecules can be targets. I'd like to introduce several applications. So if you want to test cell signal transduction, you can use LUCPorter cell lines. For example, modulation of step 3 phosphorylation by agonist, antagonist, or inhibitors can be evaluated. You can use step 3 LUCPorter cell lines. Step 3 LUCPorter cell line can be activated with IL-6 in the presence of unknown compounds or inhibitors, agonists, antagonists, for 6 hours. In this case, we used pis-ethanol, curcumin, pervanadate. Pis-ethanol and curcumin are known step 3 phosphorylation inhibitor. And pervanadate is a tyrosine phosphatase inhibitor, subsequently enhancing kinase activity, so inducing STAT3 phosphorylation more. So we quantitate LUCPorter's assay, and then we analyze the STAT3 phosphorylation pattern later. The cell line was activated with IL-6 in the presence of different concentration of PC ethanol and curcumin, and showing that the increasing amount of PC ethanol curcumin inhibit the STAT3 phosphorylation. However, perbenadate, which is tyrosine phosphatase inhibitors, enhancing STAT3 phosphorylation because that effect enhanced kinase activity. So there should be more STAT3 phosphorylation, increasing amount of perbenadate. If you have a ligand to test, you can use our LUCPorter cell line, like for example, activity of unknown ligands or agonists, as well as their EC50 values can be evaluated. For example, TLR4-ND2-CD14 IL-8 promoter LUCPorter cell line activated with LPS at various concentrations of ligand for 6 hours. And you can analyze those response assay, increasing amount of ligands. And you can also evaluate EC50. If you want to analyze like a specific toll-like receptor agonist ligands, if you have unknown ligands for TLR receptor, you can use our TLR-based NF-kappa with SEAPorter cell lines. We tested for 96-cell plate format. So each SEAPorter cell lines, such as TLR2, we tested with PAM3CSK as a control, TLR3 poly-IC as a control, and TLR4 LPS, TLR5 flagellin EC50 evaluation, and TLR7 SEAPorter cell line using rescue mode EC50 evaluation. Also same thing, TLR8 used the same rescue mode. And TLR9, we used the CPG for EC50 evaluation as a control. If you have inhibitory peptides or compound or small molecules to be evaluated, you can use LUCPorter cell lines. This was one example. TLR4 cell lines. We used Viper. Viper is TLR4 signaling inhibitor peptide, and together with CP7, which is control peptide, you can analyze dose response activity of the inhibitory peptide, increasing amount of peptide, show inhibition of TLR4 activity. And you can evaluate IC50 determination and Vipers and CP7. You can get an IC50 value. If you have an antagonist to evaluate, you can also use our LUCPorter cell lines. For example, if you have like a TLR9 specific inhibitor, like an endosomal TLR9 inhibitor, so you can compare like a TLR2. TLR2 is surface TLR. For example, if I use quinacrine, which is endosomal TLR inhibitor, as you can see, quinacrine only inhibits endosomal TLR9, and quinacrine does not inhibit the surface TLR2. TLR2 is nothing to do with endosomal acidification, so quinacrine doesn't affect TLR2 function. You can use our TLR stable cell line for TLR expression detection and calibration For instance, if you have a flow-specific antibody, TLR antibody, you can use our TLR stable cell lines. We tested several TLR stable cell lines using our TLR specific antibodies, flow-based antibodies, P-labeled PICI, and surface flow for TLR2 and TLR4, and intracellular flow for TLR3 and TLR9. So you can evaluate antibody through the flow analysis, surface flow and intracellular flow for TLR3, and intracellular flow for TLR9. Of course, you can do drug target screening using our report cell lines. If you have drug target compounds, small molecules, peptides, or natural products, you can use. For example, if you want to screen NF-kappa B inhibitory peptide, inhibitory compounds, you can use like NF-kappa B SEAPorter cell lines. We have a mock test. We have 63 different compounds, is putative NF-kappa B inhibitory compounds, and we used curcumin as a positive control. Curcumin is a NF-kappa B inhibitor peptide, inhibitory compounds. NF-kappa B activity was induced by PMA for 24 hours for the analysis. For the primary screening, those 63 compounds, you can set up like a threshold range based on the positive control. In this case, we use positive control curcumin. And based on these threshold levels, you can select several potent compounds, and you can do next secondary screening, and also you can do IC50 evaluation as well. If I summarize, you can use our cell lines for studying cell signal transduction pathways. You can identify ligands or agonists. You can identify inhibitors or antagonists. You can identify neutralizing antibodies. You can utilize, specific TLR expression detection for like a TLR detection and calibration using our TLR specific stable cell line. Of course, you can use our report cell lines for screening drug target compounds. We have screening services that called Novus Biologicals Drug Discovery Screening Services. We provide cell-based assay services, signal transduction pathways, and we also provide flow cytometry, Western, or ELISA assays. And then we also provide report cell-based compound screening services, primary screening, secondary screening, also EC50 or IC50 determination. And also we have primary cell-based assays utilizing human PBMCs, dendritic cells, B cell, and T cell subset. We can also provide Th1, Th2, and Th17 proliferation assays too. And then we have various tumor cell lines based assays. I'd like to introduce several case studies we have done earlier. And first of all, case study 1 is the proposed task was to determine whether the anti-tumor activity of the test compound is attributable to inhibitory effect on step 3 activity. What I'm trying to show in here is oversimplified and just a small portion of the whole project. And the material we used test compound, unknown compounds, and we use IL-6 and anti-phosphostep3 antibody, anti-step3 antibody, and we used the primary cells, and also we used various tumor cell lines. Of course, we use step3 LUCPorter cell lines. Our initial test for checking whether the test compound really inhibit the step3 activity, step3 phosphorylation, we checked by Western blotting, and using the primary cells stimulated with IL-6 in the presence of test compound 1 and 10 microgram per ml and different time points, and we found out test compound showed suppressive effect on step3 phosphorylation, and then the follow-up confirmation with the reporter cell lines. We checked the test compound inhibit step3 activity based on this step3 cell-based assay. And we also checked various tumor cell lines, whether test compound inhibit the phosphorylation of step3. These tumor cell lines, with the tested cell lines, we selected that these cell lines constitutively activate step3. So there are basal level of step3 phosphorylation always there. And then we treat the test compound different concentration, and then we found that the test compound suppresses step3 tyrosine phosphorylation and specifically affect the phosphorylation of step3 and several tumor cell lines. Second case study, the proposed test code was to validate and productize VPR66, a putative inverse agonist for ROR proteins. VPR66 is a proprietary compound provided by Visionary Pharmaceuticals and is proposed inverse agonist for ROR proteins. And VPR66 has been designed through the molecular modeling study using the binding sites by Visionary Pharma, which is hypothesized to bind to the ligand binding domain of ROR protein. This is a generic structure of ROR proteins. There is a DNA binding domain which recognizes specific promoter response elements, and the ligand binding domain which binds ligands. And there is a AF2 domain which is activation function 2 domain comprised of helix 12. Helix 12 binds coactivators bringing transcriptome assembly to induce the promoter activity. This image shows molecular docking of VPR66 to ligand binding domain of ROR gamma T. VPR66 is hypothesized to bind to the ligand binding domain of ROR gamma T. And this binding induces conformational change of this ROR gamma T. First of all, ROR gamma T helix 12. If I explain the hypothesized mechanism of action of inverse agonist, in normal stage, ROR protein, for example, ROR gamma T, can bind to like a specific promoter, such as IL-17A. the ROR gamma T DNA binding domain binds the promoter region, and then helix 12 binds co-activators and bringing transcriptome assembly into the promoter region and inducing the specific gene expression, the IL-17A protein expression. In the presence of ligands, like inverse agonist ligand, the ROR gamma or gamma T, this protein still recognize the promoter region. However, the inverse agonist binding to ROR ligand binding domain cause conformation change of this ROR protein, and then helix 12 no longer functions correctly and inhibiting the protein expression. And first of all, we check the VPR66 activity through cell-based assay, and we used the two different cell-based assays side by side, and one is performed by Visionary Pharma using GARPO LBD LUCPorter assay using CHO cells. This system uses transfection, transient transfection assay, transient transfection using two different plasmid vectors. One is for GARPO response element fused to luciferase gene, and the other one is ROR gamma T or ROR alpha ligand binding domain fused to GARPO DNA binding domain. And we used our LUCPorter cell line, IL-17A promoter LUCPorter cell line, and use is intact ROR gamma T protein for inducing this IL-17A promoter activity. And using two different cell-based assay, IC50 of VPR66 evaluated, and then ROR alpha, ROR gamma T, IC50, and ROR gamma or ROR gamma T IC50 evaluated. So if I summarize the evaluation, like ROR alpha and ROR gamma T, IC50 evaluated with two different assay, IL-17A promoter assay and GARPO LBD assay, showing similar results. And then later we productize this VPR66. Next case study, the proposed task was to screen the inhibitors, suppressors for IL-17A induction. We have a small library, natural product library, small molecules, and then using the IL-17A promoter LUCPorter cell lines, and also control LUCPorter cell line as a control induction. IL-17A LUCPorter cell line is inducible LUCPorter cell lines as well as semi-constitutively active cell lines. So when you compare control LUCPorter cell lines, there is always constitutive activity in there, going up to 40-fold induction. And the method was that IL-17A promoter LUCPorter cells were treated with 5 and 50 micromolar test samples for 16 hours, and then LUCPorter's activity was then analyzed. For 5 micromolar samples, you can see the green bar is at induction levels, and then orange, yellow bar is the control vector cell line levels. You can see up to 40 induction. And 5 micromolar samples, not many compounds show the activity, inhibitory activity, except one compound. And at 5 micromolar concentration, and there are several inhibitory compound selected. So through this primary screening, the natural compound against IL-17A induction yielded several potential inhibitor molecules, among which celestrol, promethazine, and thymoquinone were further selected as IL-17A induction suppressors through the follow-up test. The follow-up test we did, there are 2 different follow-up tests we performed. One is IL-17A induction IC50 evaluation for 3 compounds we selected using same cell line setup, same procedure, and we did a different concentration of each compound. And then we found IC50 for celestrol, and then dose response activity, and we also evaluate IL-17A inhibition for promethazine, and also for thymoquinone IC50 evaluation, dose response activity analysis. And then second follow-up study we performed using the human PBMC. We just wanted to check whether these 3 compounds really inhibit IL-17A production. So we checked the IL-17A induction production by ELISA. The method was that PBMC were stimulated with anti-CD3 and anti-CD28 in the presence or absence of the test samples, 3 different concentrations for 3 days. IL-17A was then measured from the cell culture media through human IL-17A ELISA assay. When you stimulate PBMC with anti-CD3 and anti-CD28, the PBMC induce IL-17A. So when we check the ELISA, indeed, celestrol, promethazine, thymoquinone inhibit, suppress IL-17A production in PBMC. The data summary, like the PBMC that were stimulated with anti-CD3, CD28 produced IL-17A, of which induction was inhibited by the IL-17A inhibitors. And then the vehicle control was DMSO, and near means no anti-CD3, CD28 stimulation, PBMC. And if I summarize the result, IL-17A induction, IL-17A production, the celestrol, promethazine, and thymoquinone, IC50 values evaluated 3 different ways. And then the human PBMC, IL-17A production inhibited by 3 compounds as well. And later we productized these 3 compounds as IL-17A induction inhibitors. If I summarize report cell line application in drug discovery, you can use target identification using our report cell lines. You can do primary hit screening and secondary hit screening, IC50, EC50 determination. And also you can do target validation, hit to lead development and lead validation and optimization. There are many steps to drug discovery goal, and Novus Biological drug discovery screening service try to focus on the first 3 steps, like from hit identification to lead optimization. We provide the screening service through several steps. Like if I divide it into the steps, first of all, you can contact Novus Biological screening services through phone or the following the email address, or you can directly visit www.drugdiscoveryscreening.com, and then we can assure confidentiality, and we can exchange NDA, and we can discuss overall project, including project scope, timetable, objectives. Then we can provide statement of work, including systems, assays, and reagents planned, data analysis format, and requirements, and timetable and milestones, and also we provide quotations. And later we do in-depth discussion with customers, and then project review, and then we review and revise SOW as agreed upon. And if clients agree the project, then we go ahead, start project, and then we present reports, and then follow up with the customer for discussing the project results. Thank you so much for attending this webinar. And if you have any questions, please let us know. Thank you. There is one question. Can I contact you if I have a target not covered by your existing cell lines? Yes, you can always contact, and we can discuss for this. Okay, there is another question. Are these services available worldwide? Yes, of course, we provide service worldwide. And in the past, we have UK, and we have Germany, and we also have Switzerland customers and Canada customers. Yes, of course, yeah, we provide worldwide. Of course, we have Asia area, including Korea, Japan customers we have in the past. Another question, what is your typical turnaround time for your assay services? It depends on the project. It can be a week to several weeks or up to 6 weeks, depending on the project and the scale of the project. So we can discuss. For the IC50 or EC50 evaluation or primary screening setup, it can be very short, 2 to 3 weeks, and or some depending on the amount of target compounds, it can go up to 1 week through 2 to 3 weeks. And another question, can Novus accept screening services using primary cells, animal, human, other than cell lines? Yes, we have primary cell-based assays. We have such as like Th1, Th2, or Th17 polyperson assay. We can analyze like if you have potential inhibitory compound for those polyperson assay, proliferation cell lines, then we can analyze those too. If you have more questions, then let us know. You can email us and you can call us, and then we can answer whatever your questions, and we can discuss with you. And again, thank you so much for attending this webinar. Then we close the webinar now. Thank you.